Okay, guys, this is Mrs. Heisenbuttle, and today we are talking about genetics, the science of heredity. You can use your science book and follow us along on pages 108 to 141 if you want to. However, if you don't have your book, or honestly you don't need it if you've got this presentation, then you can just go ahead and fill out your packet from here. So, first things first, what is heredity? Heredity is the passing on of a physical trait uh, from one generation to the next. So, a physical trait that is passed from one generation to the next is heredity. Now, the next question, of course, is what's a trait? A trait is a physical characteristic. For example, eye color would be a trait, so would height, or if you were a pea plant, it might be flower color. Those are all considered traits. Genetics is the scientific study of the passing of traits from one generation to the next. So it's the scientific study of heredity. Now, Gregor Mendel is the gent in the picture. He's an Austrian monk. That's Austrian, not Australian. And he raised pea plants, and he did all sorts of scientific experimentation on them in order to prove a lot of the things that we now take for granted as being genetic fact. So, for that reason, he's generally called the father of genetics. So, Gregor Mendel crossed pea plants that had different traits. The illustrations below show how he did this. So, the very first thing he did was that he removed the pollen producing parts of the pink flower, and then he collected the pollen from a white flower on a paintbrush and used it to paint it onto the female parts of the pink flower. So that he could have an official cross. And then the egg cells of the flower were fertilized, and they produced a new offspring. So this is how he made sure that one plant was really crossing with another. In all of Mendel's crosses, only one form of the trait appeared in the F1 generation. However, in the F2 generation, the lost form of the trait always reappeared in about 1/4 of the plants. So, if you take a look at the picture, he's got his P1 generation, which is one tall and one short. So, he crossed two purebred tall stemmed plants, or one tall stem plant with a purebred short stem plant. And then he had the first generation of offspring, which were all tall stemmed plants, which you can see in the picture. Then he allowed that generation, this F1 generation, to self-pollinate, which gave us the F2 generation over here. In that F2 generation, about 75% of those offspring had tall plant type, and 1 out of 4, or about a quarter, 25%, would have short stem type. So it just led him to believe that something was going on, because that trait, well, he thought he had bred it out of the plants, actually would show up again. And so that led him to do a lot more experimentation and led us to the idea of dominant and recessive alleles. So, a gene is a genetic factor that controls a trait. The different forms of a gene are an allele. An organism's traits are controlled by the alleles it inherits from its parents. Some alleles are dominant and others are recessive. Dominant alleles are the ones whose traits always show up. And a recessive allele is one that is hidden whenever there's a dominant version present. Mendel studied lots of different kinds and several traits in pea plants. So as you can see, he figured out what the dominant and what the recessive alleles were. And this table is kind of showing you some of the discoveries that he made in discovering which ones were dominant and which ones were recessive, which led us to a hybrid organism has two different alleles for a trait. For example, the white rabbit has two recessive alleles, and the black one has either two dominant alleles or one dominant allele and one recessive one. Geneticists often use letters to represent the alleles to make things easier. For example, capital B would be the black fur allele, while lowercase b would be the white fur allele. So a white rabbit would have to be little b, and a black one could either be big B, little b, or big B, big B. Either way. Two useful terms that geneticists used are phenotype and genotype. An organism's phenotype is its physical appearance or visible trait. An organism's genotype is its genetic makeup or allele combinations. When an organism has two alleles that match, in the example of little b, or big B, we call that homozygous. Homo means same, zygous means alleles. When an organism has two alleles that are different from each other, like one big B and one little b, we call that heterozygous. Hetero means different, zygous means alleles. If tall, capital T, is dominant over lowercase short, then a pea plant that is big T, little t has what phenotype? That's right, that would be a tall phenotype. What would be its genotype? Yes, the genotype would be big T, little t. And then what is it homozygous or heterozygous? You've got it. So that's heterozygous because it has one allele of each. Now, based on your traits inventory that you did in class last week, you should be able to answer these questions. So based on that traits inventory, what percentage of people in our class had detached earlobes? So to figure that out, you're going to take the number of people who had detached earlobes and divide it by the total number of people in the class. That will give you a number less than 1. You then multiply it by 100 to get a percentage, and that is the percentage you write on the line. So go ahead and do that for each one of the traits that we looked at last week. Okay, so the diagrams that show how to make a Punnett square. In this cross, both parents are heterozygous for the trait of seed shape R. Capital R represents the dominant round allele, and lowercase r represents the recessive wrinkled allele. So what you have is first you start by drawing a box. Looks just like that. Then you label it because they're both heterozygous. They're both big R, little r. So you put a big R and a little r on each side. Then you fill them in. So you take the ones from the left-hand side and fill across. Then you take the ones from the top and fill down. And then you have your completed Punnett square. So as you can see, if you cross a big R, little r, two big R, little r heterozygous parents, you get 25% chance of big R, 50% chance of big R, little r, and 25% chance of little r. And you can see all of that in the box provided. That is how a Punnett square works. So now we are going to be talking a little bit more about probability and genetics with some guinea pigs. So in a genetic cross, the allele that each parent will pass on to its offspring is based on probability. This Punnett square shows a cross between a black guinea pig, which is big B, and a white guinea pig, which is little b. What is the probability of getting a white offspring, just based on the Punnett square you see? That's correct. There's 0 probability of getting a white guinea pig. So what's the probability of a heterozygous offspring? Again, you are correct, 100% chance, because notice all of the offspring have a mix of big B's and little b's. None of them match. Why are the heterozygous offspring all black? Most of you have probably already figured this out. It's because big B, or black, is dominant over white, the little b, which is recessive, meaning that all those offspring have one big B, so they are all going to be black guinea pigs. Now, Mendel allowed several F1 pea plants with yellow seeds to self-pollinate. The graph shows the approximate numbers of the F2 offspring with yellow seeds and with green seeds. So how many F2 offspring had yellow seeds? So go ahead and look at the graph and see if you can answer that. You're correct, it should be about 6,000. How many had green seeds? Again, looking at the graph, it looks like 2,000. What genotypes do you think the parents had? So the question is if 6,000 came out yellow and 2,000 came out green, what genotype do you think the parents would have had? And yes, drawing a Punnett square in there with your on your paper right in here would be a great spot to draw it to show what you think they would have. Okay, so an organism's phenotype is its physical appearance or its visible trait. Physical appearance or visible trait. And an organism's genotype is its genetic makeup or allele combinations. How many genotypes are there for the smooth pod phenotype? So they're giving you over here phenotype versus genotype in the table, and they want you to look at it. How many genotypes are there for smooth pods? That's right, there's 2. How many phenotypes are represented in the table total? That's right, there's 3. Which ones are heterozygous? Big S, little s would be the heterozygous ones. And which ones are homozygous? Both the big S, and the little s would be homozygous because they match. Good job. So now that you have that all taken care of, you're going to go ahead and watch this video that I've set aside for you. It's on YouTube, How Mendel's Pea Plants Helped Us Understand Genetics. Go ahead and watch that video. It's super cute because, the peas look like babies, but also it's a really good recap of everything we've done up to this point.